What I'm going to be talking about is since this was an immunology, I thought I would really do a talk that was really related to immunology, which is really my background, my previous hat before joining Roswell. I was at Dartmouth, and that was the focus, it was all immunology. So thank you very much, and we enjoy it. So the question is, and I think we all ask this question, is why do tumors progress in spite of the fact that there are a lot of CD4s and CD8 T-cells in the tumor microenvironment? And in this case, we're going to be talking about ovarian cancers, where not only do we have the tumor itself with a lot of T-cells in it, but we also have the ascites that's often bathed in T-cells as well as tumor. And in these patients, we can also look at the peripheral blood. So I think most of you know that answer to that is because the T-cells within those human tumors are anergic or hyporesponsive to activation. And so there's plenty of T-cells. If you look at an ovarian tissue, we have the lymphocytes, a lot of lymphocytes here. Here's our tumor. In just the trichrome, you can again see the tumor. And if we look at either a CD3 or cytokeratin, you can see lots of lymphocytes, lots of CD3 positive T cells, as well as the tumor. And they're really in juxtaposition. And I think if we were to look at a variety of different tumors, we would certainly see the same type of thing. In fact, if you look at it by flow and you look at EpCAM versus CD45, there's probably more leukocytes here than there are actual tumor cells. So there are quite a few leukocytes. So what we've been doing, the assay that we've been doing, is an NF-kappa B, or similarly an NFAT translocation assay. And we've used this to monitor or to quantify the activation of T cells. So I've done plenty of work looking at proliferation. We've done plenty of work looking at multimers, and I'll show you a little bit of that later on. But this NFAT, this NF-kappa B translocation assay, is really exceptional in the sense that we can incubate the cells with an anti-CD3 and a CD28 or a number of different things and see activation really within a couple of hours. So it's a relatively rapid, and it does correlate pretty well with what you would see downstream. So the translocation of NF-kappa B and NFAT from the cytosol, so from the cytoplasm, into the nucleus, we can basically measure by confocal microscopy. We've done quite a bit as well by image stream. And it's really a pretty straightforward assay. What we would do is we would bind CD3 and anti-CD28, and then what you're going to do is basically get phosphorylation, and all that is going to end up in translocation of the NFAT and the NF-kappa B into the nucleus, and so we can consequently measure this translocation of either the NFAT or the NF-kappa B into the nucleus. Looking at it by confocal, here's an unstimulated versus an anti-CD3, anti-CD28 stimulated culture, and you can see here with the CD3 standing, nice ring standing around the periphery. Then the NF-kappa B and the untranslocated around the nucleus, whereas in the one that's been stimulated, we've got nice translocation into the center of the cell. And then here's our nuclear stain, and so we can see when we merge them that this is in the cytosol, whereas we have co-localization of the NF-kappa B with basically the DAPI stain that we're seeing inside the cell. So pretty easy to distinguish between a stimulated and an unstimulated cell. I often get accused of looking at just one cell, and of course we do that a lot when you're by confocal. But if we take a back step and actually look at a field of cells, you can see that in the unstimulated we've got really a good population of T cells. Pretty much all of them show the cytosolic expression of the NF-kappa B, and if we were to enumerate it, we get about 1.5% show some form of activation. Whereas when we hit these with an anti-CD3, CD28, it's pretty clear that we've got a lot of translocation going on in those cells there. So if we, so the question is, T cells from primary and metastatic ovarian tumors are hyper-responsive to CD3 and CD28 stimulation, but responsive to other mechanisms of activation. So this is really one of the very first experiments that we did. And what we did was we took peripheral blood T cells or lymphocytes and activated those with either anti-CD3 or CD28 or cross-linked or activated them with TNF-alpha or alternatively with PMA anamycin. And what you can see is that we get pretty, this is our maximum, the TNF or the PMA anamycin is really our maximum amount of NF-kappa B translocation. And you can see you get very similarly good activation in the PBLs when you do an anti-CD3, CD28. But if you do these from patients that have an ovarian carcinoma, if you do this from cells that are taken from an ovarian carcinoma, what you can see is that, sure, we get activation and translocation of the, through TNF or PMA anamycin, but we don't see that with the ovarian carcinoma. So the question is going to be, why is that? Why do we see this lack of this hyper-responsiveness? Just in terms of looking at that assay, that was done by Confocal. We've also used the image stream for this, and I won't go into the image stream. I think Renee would like me to go more into what the Novocyte is about, but in terms of the image stream, what we're looking at here is an untranslocated versus a translocated, and we can separate that into two images, in this case with the NF-kappa B, and here with the 7-AD, and when we merge them, we get this type of response there. Now we can measure that on the image stream basically by looking at what's called a similarity score, which is basically here. And this is looking at the NF-kappa B intensity versus the 7-AD. If there's no translocation, that's going to be reversed. So you're going to have high expression of 7-AD and low expression of NF-kappa B, and vice versa. Same way here. Here we've got the translocated, and we've got good co-localization, high expression of NF-kappa B, and relatively high expression of the 7-AD. So this actually gives us a value that we can then use and plot, the 2.81. And this is for one cell. So this is not a variety of cells. This is actually the score from that particular cell up there. And of course, we can run thousands of cells through the instrument. So here's an example now where we're combining those numbers from a lot of different cells, where we've got NF-kappa B and NFAT, and you can see we get nice measurable similarity, increase in the similarity score from those. So that allows us to basically use this method to do a lot of quantitative analysis. Using that, again, we see the same type of thing, where if we incubate PBLs with either CD3 or PM anamycin, we get nice high RD value or high similarity score. If we take it from an ascites, an ovarian ascites, we really lose that ability to translocate. And if we take it from an ovarian tumor, it's even worse. So we see clearly a correlation with a lack of translocation in the tumor microenvironment. So the conclusion from all this is that T cells from within the tumor are hyperresponsive to anti-CD3 and anti-CD28. So I think we all were to step back. There's a variety of different mechanisms, and they're probably all involved in the process that cause this hyperresponsiveness. T regulatory cells, MDSCs would certainly be a popular one right now. Different suppressor molecules, checkpoint inhibitors, and that type of thing. But really what we're focusing on here is that phospholipids in the form of exosomes are released from the cells in the tumor microenvironment, and we're hypothesizing that these are in part responsible for the T cell arrest. So while I think that these are all probably involved, this is another potential mechanism. I think by the end of the talk, you'll believe that this is true as well. Okay, so if we were to take the, without the ascites, nice similarity score here. If we add ascites from these tumors to a just normal peripheral blood, you see that there is a dramatic inhibition. And that's actually titerable to some extent just by adding in more ascites and then titering it out, and you can see that you basically get that loss. So it's a titerable effect, and it clearly is, so it's associated with the ascites. So something within the tumor ascites is causing T cells to be hyper-responsive. And so the first thing that we did, or one of the things along the line that we did, was to sort the T cells and ask the question, is this just with CD4 or is this just with CD8? And so we sorted the preps, and sure enough, it turned out that both the CD3s, CD4s, and CD3, CD8s were involved in the response. So here it is unactivated. This is with PMA. Here we are cross-linking with CD3, CD28. And then when we add the tumor ascites, we see an inhibition of both the unsorted cells as well as the CD4, and CD8 fractions. So the tumor acts directly upon the CD4, or at least the tumor ascites acts directly upon both the CD4 and CD8 T cells to inhibit their activation, presumably through the TCR because of the anti-CD3. So what is the immunosuppressive factor present in the ascites? Well, that's the question for now. So we did a number of the types of experiments that you would all do just to try to begin to uncover what's causing this. And the first thing was figuring it was probably a protein, was just to heat and activate it by boiling it for 10 minutes. And that really had no effect on it. It was also resistant to proteolytic enzymes, even after prolonged exposure to pronase. It did, however, partition into the organic phase when we did a hexane isopropanol diffraction, and the same thing happened with ether. And so this began to make us think, well, it could be a lipid. So we have, we basically took a delipidated prep, incubated these with the anti-CD3, CD28, and what you can see is that if it's unactivated, we're here. The CD3 is here. If we just add in the ascites, we get a partial inhibition. But if we delipidate it, then that activation is returned to normal. So it does appear to be in the lipid fraction. So the inhibitory factors in the tumor ascites partition into the organic phase and is likely some form of a lipid. So I have a friend, actually a person that I've collaborated for many years, by hook or by crook, is Charlie Berenson, who's at the VA in Buffalo, and he does a lot of work. This is definitely not my area with lipids. It's CDs and flow cytometry, but he's done a lot of work with these. And we basically came up with a list of what was there. Now there was a manuscript by this also guy who basically had shown in jerked T cells that after activation with PMA, that they had inhibition to cross-linking with either CD3 or CD2 of the phosphatidylserine synthesis. So for this and a variety of other reasons, we really began to think about phosphatidylserine as something that we wanted to explore first, as opposed to going through the whole list. Although at this point, we're actually now beginning to look at some of the gangliosides. So what we did was we got together with Sathi Balu Iyer, and he made a number of liposome preparations for us. And one of them was just phosphatidylcholine, and then the other one was phosphatidylserine and phosphatidylcholine. You can't just use phosphatidylserine because those liposomes will aggregate and you get into a lot of problems. But so this was a combination of the two. And what you see is that if we use the phosphatidylcholine, very little or any inhibition, certainly not significant. Whereas if we had a component that had the phosphatidylserine, those were in fact inhibitory. This was titerable, so at higher levels of phosphatidylserine, there was more inhibition than at the lower levels. So the conclusion was phosphatidylserine by itself has the capacity to arrest at the activation of T cells. In what form then is this phosphatidylserine being expressed? And so the options are soluble, micellar, liposomal, exosomes, apoptotic vesicles. lipoprotein complexes, and possibly others. So what we did next was to basically do a ultracentrifugation. And here what you're seeing is with the ascites, our inhibition, in comparison to our activated with anti-CD3 and CD28. And then here is where we take the ascites fluid that's been ultracentrifuged, and we're just taking the supernatant. And so it was something in the ultracentrifuge pellet that was responsible for this activity. So inhibitory phosphorylation is present in a vesicle exosome that pellets with centrifugation. If you take a look at that pellet, what you can see is a lot of vesicles. These are exosomes, basically just an awful lot of them. And these had a distribution of, well, on average 30 nanometers, but a distribution between 10 and 100 nanometers. If you were to basically add those exosomes back in, we see that it causes inhibition. So here's with just the ascites fluid in comparison to our activation control. Here's with the exosomes, and then here's with exosomes plus an anti-phosphatidylserine antibody. So we can see that if we just use ascites or exosomes, we get inhibition. But then if we come in and we add the exosomes and we block the phosphatidylserine with an antibody, we're seeing an increase. So it's clearly on the exosomes, and it's clearly related to the phosphatidylserine. Now, my wife seems to think that I've been talking to a good friend, Dick Bankert, and I'll show you some more of his data in a second, and we'll be at the dinner table, and she just doesn't get why we are so interested in these microvegetables. And so she created this slide for me, trying to describe what, in fact, the microvegetables were, at least to her. But here's really what I think of them as. And this is some early work by Dick, a good friend, where he's basically showing these vesicles that are being produced. In this case, it was a B-cell lymphoma, but these vesicles that are produced by the tumor cells. And it's really fascinating as you get into the literature with this about what these things are capable of doing, just in terms of communicating from one cell to the next. If you have like a GFP mRNA in one of these vesicles, you can see it then attaching to another cell that doesn't express the GFP and lighting up, so you can have exchange of proteins, exchange of message, exchange of DNA. So there's really probably a lot of communication that is going on with these things. They're basically two mechanisms of production. The ectosomes, which tend to be bigger, these are here. These are being produced by budding off from the membrane, and they could contain a variety of different things. And then these exosomes, which tend to be much smaller, in the 30 to 100 nanometer range, and these are usually formed in vesicles, and that then bind with the membrane and then are expressed. So in a vesicle that binds with the membrane and then are expressed. And again, you can see them here, and also with some dyes here. We've been interested in them for a variety of reasons. Ultimately, what we're trying to do is develop some assays to look at minimal residual disease in patients with tumors. And with the idea being that we could detect them in the blood and then use that as a bioassay. So we've done some phenotypic analysis of the exosomes by flow. And this is really a difficult thing to do. We're trying to look at particles that are very small. As I point out, we're talking about 30 to 100, maybe 200 nanometers, which is really at the limits of what you can do by flow cytometry. But our analysis reveals that there is basically phosphatidylserine on the surface of the membrane. So we've done some work with this with the Novocyte, and what we've been running basically to get it to sort of standardize is these apogee beads. These are sizing beads, and they range from 0.1 micron, 100 nanometer, all the way up to 1 micron. There's two of them that are fluorescent. So there's one up here at about 500, 0.5 microns or 500 nanometers that is fluorescent, and then another one here that's really right at about 100 nanometers. What we can do, and actually this is something that Renee and I have been talking about in terms of a white paper, so it's something that we'd be able to share with you in the long run, is develop a methodology to detect these on the NOVA site. And the strategy is really to clean up the buffers. So normally these are going through like a 0.2 micron filter set. So the strategy is really to try to get those purified at like a 0.1 or lower process, and then to basically set up some gating. Often it's good to do that on your scatter off of the 405. What I'd really like to do, and what we've been exploring is vesicles that are being produced by a cell line that has GFP in it, and so we can then clearly identify, detect that this is a vesicle. It's not noise that's in the instrument. And then looking at different dyes, and for that we've been playing around with actually this cell trace file dyes work very well as the more lipophilic dyes. Lipophilic dyes require an ultracentrifugation, but the tracking dyes, the CFSEs, the cell trace violets, those you can really utilize without a wash step, which is important when you're talking about very small things. So this is really where we've been going with the NOVA site that we have at Roswell. So here's some data with the basically looking at phosphatidylserine. It's just coming in with an Nexin 5 that has phycoerythrin on it, and you can see it's clearly that a portion of that is at least a portion of this preparation is expressing phosphatidylserine. This is work that Joe Tari at the lab did, and it's blockable. So I think you could look at this and you could say, well, what is that? Is that real or is that not? But the fact of the matter is that if we come in with a cold, an unlabeled Nexin 5, it's blocked. But it's not if we use a same concentration of BSA or IgG. So I do think it's real, and that's when you're down at this level, it's very hard to convince yourself of that. So we concluded that phosphatidylserine is definitely on the present of the exosome surface, and we've gone on to do some phenotyping of that. These are by other measures exosomes. Here's a positive control, basically a Western array. This is the negative control. And then you have a number of the tetraspanins, CD81, CD63, many of the different things that you would characterize an exovesicle with that are basically being expressed by these things. Gotham, who is in Dick's lab, has gone on to take this beyond the NF-kappa B translocation aspects of it. So one of the issues, one of the reviewers were concerned that, well, you're just looking at NF-kappa B. Why don't we talk about things that are further downstream? And so here we're looking at CD4 and CD8 cells, and then this is upregulation of CD69. So controls, very little activation. If we hit them with the anti-CD3 and CD28, you were finding that almost 90% of the cells express CD69. And then if we throw in the exosomes, that's reduced significantly down to 32 in the CD4 cells or 28% in the CD8 cells. Same type of thing is true with CD107A. Again, control. This is activation. And then if we throw in the exosome pret, it's almost completely, if not completely, inhibited by the exosomes. So clearly an example of not only the NF-kappa B and fat translocation, but further downstream effects. But I can hear you want to go further downstream. So here we have with looking at IL-2 and interferon gamma production. And you can see that with just the CD3, CD28, we're seeing some activation of the 4s and 8s. and that is essentially blocked when we throw in the exosome prep. Same type of thing here. When we're looking, this is going to be with IL-2, and here it is with interferon gamma, where we're going from a 0.2 to a 6 or 9%, and then dropping back down to a 1% in the presence of the exosomes. So we've also done some work with this, looking at antigen-specific expression. So here we have work that's been done on the image stream, looking at CD3 cells here, and then taking those into basically the CD8 population. And now I had explained to you before the similarity score and basically how that worked. So now we're looking at basically each one of these dots as a cell, and we're looking at the NFAT similarity score. This is on ones that are multimer negative, and this is on ones that are multimer positive. So what do I mean by that? Well, we incubated the peripheral blood leukocytes with an EBV multimer. And that's going to bind to the T-cell receptor, and then really it's within just a question of 2 hours that you will see the translocation of the NFAT as measured by the image stream. So it's interesting, and this is a whole other talk that we've sort of taken into transplant patients. But it turns out, if this is our negatives, that there's clearly a population. And if you look at enough of these plots, you believe it, that of T cells or CD8 T cells that are, even though they're capable of binding the EBV multimer, they do not translocate. They're anergic. They're essentially anergic. Whereas there's another population that is not. And so that's been an interesting finding. And it's actually been a really interesting assay to work up. So we've done that now with our exosome preps. And what you can see here is after 2 hours of incubation, either on ice or at 37 degrees, that in the presence or the absence of, in the presence of the exosomes, we have significant inhibition. Really, it's the same as incubating the cells on ice. Whereas for the vast majority, if we look at the individual similarity scores, we have quite a good level of activation in the absence of the exosome. So we're really seeing this with the NF-kappa B translocation, the NFAT, CD69, CD107A, the cytokines, and now not just hit them over the head with an anti-CD3, CD28, or PMA. We're actually seeing this with a specific type of immune response. So we really do believe that these microparticles, these EVs, are responsible for at least some of the inhibition that we're seeing in the tumor microenvironment. So inhibition of CD8 T cells with tumor-associated exosomes inhibits an antigen-specific T cell response. So what is the mechanism by which exosomes inhibit T cell signaling? This is just preliminary data. This is stuff that we're looking at. But basically, the idea is that a diacylglycerol kinase, DAG, is very sensitive to or responsive to phosphatidylserine. And the thought is that as the phosphatidylserine enters into the system, that upregulates the activity of the DAG, and what that is then responsible for is the inactivation of the phosphatidic acid, and that's really going to be one of the very first steps in terms of basically either the NF-kappa B or the NFAT in terms of that cascade. So that's what we're working on now, and actually, if we, again, preliminary data. But if we show, if we look at if we inhibit the diacylglycerol kinase with this, what we find is that it reverses that TCR signaling. So here, not activated. Here's the CD3, CD28 activation. Throw in the ascites, and then we have this inhibitor of the diacylglycerol, and our activation is basically increased, and it really doesn't do anything for the p-aminomyosin. So that's really where we're working on now and where we're headed with on this project. So phosphatidylserine present on exosomes induces the activation of diacylglycerol kinase in T cells, and then converts the diacylglycerol to, it converts it to phosphorylic acid, thereby blocking the TCR signaling. So the conclusions from this, our overall conclusions, are that exosomes present in the human tumor microenvironment inhibit T cell function. Phosphatidylserine present on the exosome surfaces induces a rapid and reversible T cells arrest of T cell signaling. Exosomes are derived from the tumor cells as well as non-neoplastic. I didn't show you that data, but we can also show that there are T cells and some epithelial cells that are producing the at least have phosphatidylserine on their surface by flow cytometry. And the phosphatidylserine binding induces the activation of diacylglycerol kinase, resulting in a blockage of downstream signaling by DAG. Where do you want to go with this? What do we want to do next? Well, the question is, can we come up with a way of blocking this in vivo? So is there a way that we can basically inhibit the anergic effect of the phosphatidylserines and apply this in our immunotherapies? Are there differences between different subpopulations of the exosomes in terms of their ability to inhibit or enhance tumor? We obviously do more on the mechanisms by which the exosomes inhibit T cell activation, and then what mediates the actual release of exosomes from a tumor cell. Is this just an ongoing, or is there something that's very specific to it? So I would like to. This is really I'm just a small part of this project. It's been one that I've been involved in, but over at UB, Dr. Richard Bankert, Dick Bankert, dear friend and colleague, has really gotten me involved in this and gotten us all interested in it. Safi, I mentioned him. He was involved in making liposomes, which is one of the steps that we're using to sort of validate our approach, long-term validation of being able to measure these by flow. Gautam did a lot of the work that you saw with a lot of work just in terms of purification of these preparations, but also work in terms of the downstream signaling, the IL-2, interferon gamma, C107A work. And then I mentioned that the group at Roswell, Kunle Adunsi is the surgeon responsible and has provided us with all the preparations for the ovarian. And Joe and Hans and Orla have all been working on various aspects of it. Anyway, this is a group at Roswell. And for that, I thank you very much for your attention and would be happy to take any questions.